Hello and welcome to Chapter 28 of Biology in Focus, Plant Structure and Growth. This is for John Tyler Community College, Biology 102, General Biology 2. My name is Mr. Sparks and I will be taking you through this lecture. Okay, well, here's a Romanesco broccoli. It's you can get these in certain markets, probably like Elwood Thompson's or other fancy food markets, Trader Joe's or what have you. And it's just a type of broccoli, but it shows a particular growth pattern associated with a mathematical formula known as a Fibonacci series or a fractal pattern. This is just a way that this is a way that most plants grow, but most plants are more subject to their environment, and so although they grow from a particular point, the apical meristem, which is the point on each one of these little areas of growth here, this is the apical meristem, they grow in a particular pattern when the conditions are favorable, and which they often are for this particular type of broccoli. Okay, so each of its smaller buds resemble a miniature of the entire vegetable. Mathematicians refer to such repetitive patterns as fractals. If Romanesco looks like it was generated by a computer, it's because of a growth pattern that follows the repetitive sequence of instructions. Most plants are subject to greater diversity in their vegetative growth, but I'm not going to spend too much time describing this. If you want to look at more information about the Romanesco broccoli, you can look on page 553, which is the first page in the chapter 28. Angiosperms are primary producers and are important to agriculture. Taxonomists split angiosperms into two major clades, monocots and eudicots. Used to be called dicots. Mono means one and di means two. And eu means true, so eudicots are the true dicots. Angiosperms are the flowering plants. They're distinct from the gymnosperms. We learned about this differentiation in the last chapter. Angiosperms are mostly what we think of. It's everything except for pine trees, basically. And there's more detail than that, of course. You know, there's the bryophytes and the gymnosperms, but angiosperms are the primary producers and are very important in agriculture. Okay, here's a table of characteristics of monocots and eudicots. These classes of angiosperms are named for the number of cotyledons or seed leaves that they typically possess. Monocots typically have one cotyledon, and eudicots have two cotyledons. Monocots include orchids, bamboos, palms, lilies, as well as grasses such as wheat, maize, and rice, and corn. A few examples of eudicots are beans, flowers, sunflowers, maples, and oaks. Okay, so here are the different traits. We're going to go through them in a little detail here. Here the monocot. This is just like a corn seed. The corn seed has one cotyledon. It's covering it's the little green leaf inside here. Here's the endosperm, which is the majority of the seed. The endosperm provides nutrition for the seed to grow. Here's a eudicot seed, like a bean. This has two cotyledons. You can see here these primitive or these tiny little leaves inside the endosperm, inside the shell of the seed. And monocots usually have parallel venation. You can see this pretty readily on the leaves of orchids, or if you've ever seen corn, if you've been driving through the country and you step out of your car and into a cornfield, you can see the corn leaves they have parallel venation like this. Grass is another easy example to see. If you just pick up a blade of grass, you can see that it has a parallel venation. it is a monocot. Eudicots usually have net-like venation or reticulated venation. This is the leaf of a sweet gum tree. You know, sweet gums are those trees that drop those little spiky balls all over the ground. They're really kind of annoying, and they're not really great firewood. You can't really build anything out of them because of the way that they grow. It's not a very useful tree. Monocots have vascular tissue that's scattered within the stem, and eudicots, the vascular tissue in the stem, is more organized. Monocots typically have a fibrous root system that spreads out across and locks it into the soil, and eudicots typically have a tap root, like a carrot is a eudicot that has the tap root, and the tap root of the carrot is the carrot itself that we eat. So that tap root is usually present in eudicots. Monocots have a pollen grain with usually has one open, and eudicots have a pollen grain with 3 openings. And the flowers are distinct on monocots. They usually have multiples of 3, so they can have 3 or 6 leaves, like a lily or an iris. Any one of these, any one of the springtime flowers. Daffodils have a fusiform umbel, but it does have a 3 set of 6 leaves surrounding that function. And floral organs in eudicots usually occur in multiples of 4 or 5. So, like an apple flower, or even a rose is a eudicot, but it's harder to tell because it's got so many. The form of the rose it really contains many mutated sepals and more numerous petals. It's been bred for that condition. But eudicots usually have multiples of 4 or 5 in their flower petals. Okay, plants have a hierarchical organization constituting of organs, tissues, and cells. Plants have organs composed of different tissues, which in turn are composed of different cell types. An organ consists of several types of tissues that together carry out a particular function. A tissue is a group of cells consisting of one or more cells that together perform a specialized function. So plants have these hierarchical structure of organs, tissues, and cells. There are 3 basic plant organs: roots, stems, and leaves. The basic morphology of vascular plants reflects their evolution as organisms that draw nutrients from below the ground and above the ground. Plants take up water and minerals from below the ground, and plants take up carbon dioxide and use light for photosynthesis above the ground. So they produce carbohydrates above the ground, which they transfer around the plant and through the phloem, the vessel that transports food, and around into the plant. And the water and minerals comes from the opposite direction. 3 basic organs evolved to acquire these resources: roots, stems, and leaves. The root system includes all of the plant's roots, basically everything that's below the ground. The shoot system includes the stems, leaves, and in angiosperms, the flowers. So this includes everything that is above the ground. Okay, here is a schematic of an overview of a flowering plant. The plant body is divided into a root system and a shoot system connected by vascular tissue, which are the purple strands in this diagram. It is continuous throughout the plant. The plant shown is an idealized eudicot. Okay, so you can see it has netted venation in its leaves, and it has a 5-part symmetry in its flower. It's a eudicot. And here are some different parts of the plant that we're going to describe in this chapter. All right, here's the root system down here, the shoot system up above, everything above the ground. The reproductive shoot, this is the reproductive organ, this is the flower. The apical bud is this tiny point here. It's at the top of the plant. It's the vegetative part that continues to grow. It's known as the apical meristem or the apical bud. Here is the node. The node is where the leaves or the flower come off the central portion of the stem, the central stem of the plant. The internode is the area between nodes. So this is a node here, and this is a node down here, and this is an internode here. And here is another apical bud on the tip of this stem of this flower. Okay, so this is another apical bud. And here's the vegetative shoot, which is a stem, or will become a stem or a branch if the plant grows into a tree. Here is the leaf of the plant, which includes the blade, which is the large flat part of the leaf, and the petiole, which is the stem that attaches to the main stem or the trunk of the plant. These are the axillary buds. Axillary buds are generally non-vegetative, but they can give rise to additional stems. The additional stems will grow in those nodes, and they will become another place for flowers or leaves to branch out. Okay, here is the main stem, which provides the main structure of the plant, and then below ground we have here the taproot. The main function of the taproot is to secure the plant into the soil, and then lateral or branch roots. And these lateral branch roots grow underground here, and they also have little apical meristems that grow throughout the soil and increase the surface area of the plant, the ratio of the plant to the soil. This is where the water and the minerals comes in through the base of the plant, and then up through the stem into the leaves, and then all the photosynthetic function is occurring in the leaves, and the carbohydrates are being produced, and they're going back down into the rest of the plant, transferred through the vessel system. Roots rely on sugar produced by photosynthesis in the root system. Okay, so the roots, in order to grow, they need photosynthates, carbohydrates and whatnot from the shoot system. And shoots rely on water and minerals absorbed by the root system. A root is an organ with important functions. It anchors the plant, it absorbs minerals and water, and it stores carbohydrates. The roots of the plant in a perennial plant or a biennial plant, a plant which comes back every other year, or a perennial plant comes back every year, they may store carbohydrates in that plant, and the stem and everything above the ground can die off, and then that plant will come back the next year, just like daffodils in the spring. Tall, erect plants with large shoot masses have a taproot system, which consists of a taproot, which is the main vertical root, and lateral roots, the branching off the taproot. Small or trailing plants have a fibrous root system, which consists of adventitious roots that arise from stems, and lateral roots that arise from the adventitious roots and form their own lateral root structure. In most plants, absorption of water and minerals occur through the root hairs, thin extensions of epidermal cell that increase the surface area of the root. Okay, these are the root hairs of, it looks like maybe a monocot or corn. Looks like a corn kernel to me. Could be a bean. It could be a eudicot. In any case, in all plants, there's this root structure. There's a main taproot, and then these lateral roots that come off of the side. And these, in this case, in this very small root here, these tiny root hairs, they really enhance the ability of the plant to grow, to absorb water and nutrients from the soil. And this is also where the the interaction with mycorrhizae occur. If you recall, in the previous chapter, we talked about mycorrhizae and their relationship to plants and how they benefited plants to help them absorb nutrients through their root structure. Well, this is where that happens. It all happens in the fine root hairs of the plant. Most terrestrial plants form mycorrhizal associations, which increase mineral absorption. Many plants have root adaptations with specialized functions. We're going to look at some of these here. Okay, here are the pneumatophores of a mangrove swamp, the storage roots of a beet, and the strangling aerial roots of a ficus or a fig tree. Now, the pneumatophores of a mangrove swamp, if you can see in the background here, these are the mangrove trees, and they grow around the world. It's the same species that grows around the world. It grows in Florida. You can find mangroves. You can find them anywhere in the Caribbean. If you've ever been on a Caribbean vacation, you might have seen the mangrove trees, and they produce like a little. They produce partially formed offspring that drop into the water, and they get washed away in the sea, and they can wind up on the distant shores of any particular island. But these pneumatophores, which are important to the structure of the trees because the roots can't get air in the dense mud that gets trapped by these pneumatophores. So they instead on the parts of these pneumatophores are structures called lenticels, and the lenticels allow for the transfer of air between the plant and the root. The mangrove tree itself also has lenticels, but it needs the lenticels from these pneumatophores in order to be able to survive. Now, another interesting thing about this is these pneumatophores trap the sediment. So they trap all this mud and sediment that you see down here, and a mangrove can build its own island. So a mangrove gets situated in a shallow area. It begins to reproduce and drops its partially formed offspring into the soil, and then as the more and more pneumatophores occur in that area or grow in that area, it traps more and more mud, and more mud makes more and more land for more and more mangroves. So mangroves actually build islands all throughout they're low-lying islands, and they don't really you can't really live on them too well, but they help to secure the structures around these islands. And when there's a big hurricane, it strikes the mangroves, and the mangroves protect the land inland. Okay, here's a beet. I don't know if you know this, but sugar beets are very important. Most of the sugar that we get in this country is from areas like North Dakota, where they have vast amounts of tillable soil, and they grow beets there. These are sugar beets, and sugar beets store carbohydrates like sugars in that root ball, in the roots of the sugar, and in that bulb, in that tuber. And the sugar beets can be harvested, and then they're processed, and sugar cane from tropical sugar cane is also another source of sugar. But in this country, we get a lot of our processed sugar from sugar beets. So sugar beets are producing, they're photosynthesizing up in their leaves up here, and then they're shooting that photosynthate, the sugars, down into the storage roots, down into the tubers. And then here are strangling aerial roots. These are roots that provide structure. This is a famous temple in Cambodia known as Angkor Wat. Angkor Wat is like a 2,000-year-old temple, and it's been left to the mercies of the jungle. And these ficus, these fig trees that grow here, are pretty common all around the world, all around the ruins. It's a tropical fig tree, and you can see it will the fig itself will get eaten by a bird, and then it will be the bird will defecate on the side of a tree, and the fig seed will grow, and it will grow down those roots all around the structure of the original tree. A stem is an organ consisting of an alternating system of nodes, the points at which leaves are attached. Internodes, the stem are the stem segments between the nodes. An apical bud or terminal bud is located near the shoot tip and causes elongation of a young shoot. An axillary bud is located in the upper angle formed by the leaf and the stem, and has the potential to form a lateral branch, a thorn, or a flower. Axillary buds are generally dormant. Some plants have modified stems with additional functions. Okay, a rhizome is actually a modified stem that grows underground, and it gives off roots. Rhizomes are typically what we associate with the iris flower. That grows from a stock rhizome. Stolons, this is a strawberry here. This is a strawberry plant. The strawberries, when they get to a certain point in their growth phase, they shoot off these little offspring on the ends of these rootlets, and these rootlets, once they touch the ground, they begin to grow. So they have adventitious roots that take advantage of the soil, and when they land on the soil, they form a new strawberry plant. So here's the parent strawberry plant, and the stolons, which fire off on the side of the original plant. The plantlets land in the soil, and they produce their own roots once they land on the soil there. Tubers are large organs for storing carbohydrates of the plant. Tubers such as these potatoes are enlarged ends of rhizomes or stolons. They are specialized for storing food. In fact, the eyes of a tuber are the clusters of axillary buds that mark the nodes. So they're really modified stems. They are not parts of the roots, the way that a beet root is. Okay, rhizomes, the base of this iris plant, is an example of a rhizome. Its horizontal shoot grows just below the surface. Vertical shoots emerge from axillary buds on the rhizome. Shown here on a strawberry plant, stolons are horizontal shoots that grow along the surface. These runners enable the plant to reproduce asexually, and as plantlets form at the nodes along each runner. And here are the tubers of the potato. A leaf is the main photosynthetic organ of the vascular plants. Leaves have other functions, including gas exchange, dissipation of heat, and defense. Leaves generally consist of a flattened blade and a stalk called the petiole, which joins the leaf to the stem. Monocots and Eudicots differ in arrangement of veins, the vascular tissue of leaves. Most monocots have parallel veins, and most Eudicots have branching veins. Some plant species have evolved modified leaves that serve various functions. Everything we see here is a modified leaf. The spines of a cactus, the tendrils of a grapevine, the reproductive leaves of a kalanchoe, a certain type of succulent plant, and the storage leaves are modified leaves of the onion. These tendrils by which this pea plant clings to a support are modified leaves. After it is lassoed to support, the tendril forms a coil that brings the plant closer to the support. Tendrils are typically modified leaves, but some tendrils are modified stems, as in grapevines. Spines, the spines of a cacti, such as this prickly pear cactus, are actually leaves. Photosynthesis has been abandoned in the spines, and it is now carried out by the fleshy green stems. Storage bulbs, such as this cut onion, have short underground stem and modified leaves that store food. So, I'm sorry. This is the storage. These are the storage leaves. Bulbs such as this cut onion have short underground stem and modified leaves that store food. So here's the stem down here in the base of the onion. This all grows underground here, and then above ground, the photosynthetic part of the plant. It looks just like a daffodil, or you've probably seen wild garlic. It's the onion has a very simple. It's a monocot. It's a monocot. Reproductive leaves. The leaves, such as of some succulents, such as this calancho, produce adventitious plantlets which fall off the leaf and take root in the soil. So these leaves can be part of the reproductive structure of the plant. Each plant organ has dermal, vascular, and ground tissues. Each of these three categories forms a tissue system. Each tissue system is continuous throughout the plant. Okay, the dermal tissue system in blue here provides a protective cover for the entire body of the plant. The vascular tissue system here in purple transports materials between the root and the shoots of the systems. It is also continuous throughout the plant, but it is arranged differently in each organ. The ground tissue, shown in yellow, is here responsible for most of the plant's metabolic functions, such as the located between the dermal tissue and the vascular tissue of each organ. Okay, so the dermal tissue, everyone probably knows that the epidermis is your skin. So the dermis, the dermal tissue, is the outer covering of the plant here. And then there's ground tissue, which is the supportive tissue inside the leaf or inside the stem, and then the vascular tissue. Now you can tell if you look at these are two different types of plant. One is a eudicot and one is a monocot, and you can tell the eudicot has a greater structure of the vascular tissue, and the monocot is less structured. The dermal tissue is an outer protective covering. In non-woody plants, the dermal tissue system consists of the epidermis, a layer of densely packed cells. In leaves and most stems, a waxy coating called the cuticle helps prevent water loss from the epidermis. In woody plants, protective tissues called periderm replace the epidermis in older regions of stems and roots. Trichomes are outgrowths of the shoot epidermis, which can help with the insect defense. Okay, we're going to talk a little bit about trichomes in the lab and some of these structures in greater detail. Trichomes are what they often provide certain chemical compounds will occur on the tips of the trichomes, and these can help to be insecticides to defend against marauding insects. The vascular tissue system facilitates transport of materials through the plant and provides support. Two vascular systems are the xylem and the phloem. The xylem conducts water and dissolved minerals upward from the roots and into the shoots. The phloem transports organic nutrients from where they are made, photosynthesis in the leaves, to where they are needed, which is the rest of the plant and particularly the roots. Okay, so that potato is a product of the leaves of the potato plant. The leaves of the potato plant photosynthesize. They produce photosynthesis and carbohydrates and sugars, and those are transferred down under the ground into the structure of the potato, and that's where they store the carbohydrates where the plant may use those carbohydrates in the following year, or to winter over the winter. The phloem transports these organic nutrients. Now, the difference between the xylem and the phloem is the xylem transports water and minerals, and the phloem transports food. So the phloem transports food. You can always remember phloem because it sounds like food. Phloem, food. The vascular tissue of a root system is collectively known as the stele. In angiosperms, the root stele is a solid central vascular cylinder. The stele of stems and leaves is divided into vascular bundles, strands of xylem and phloem. Tissues that are neither dermal nor vascular are the ground tissue system. Ground tissue internal to the vascular tissue is pith. Ground tissue external to the vascular tissue is cortex. Ground tissue includes cells specialized for photosynthesis, short distance transport, and storage or transport, or storage or support. Plant cells have structural adaptations that made their specific functions possible. The major types of plant cells are parenchyma, collenchyma, sclerenchyma, water-conducting cells of the xylem, and sugar-conducting cells of the phloem. Parenchyma cells have thin, flexible primary walls. They lack secondary walls, and they have a large central vacuole. They perform most of the metabolic functions. They retain the ability to divide and differentiate. So, like chloroplasts occur in the parenchyma cells. Parenchyma cells perform most of the metabolic functions of the plant, synthesizing and storing various organic products. For example, photosynthesis occurs within the chloroplasts of the parenchyma cells in the leaf. Okay, here are chloroplasts inside the parenchyma cells of Elodia, which is a common water plant. If any of you have aquariums, you may be familiar with Elodia. It's a very common freshwater water plant. And here you can see the structure of the cells here. These are the parenchyma cells. Collenchyma cells are grouped into strands. They help support young parts of the plant shoot, and they have thicker and uneven cell walls, and they lack secondary walls. They provide flexible support without restraining growth. Okay, at maturity, these cells are living and flexible, elongating with the stems and the cells they support, unlike sclerenchyma cells, which we'll discuss next. Here are a group of collenchyma cells in a sunflower plant. The sunflower is a pretty green plant, and it's got these collenchyma cells running through it, and they help to support the flower of the sunflower is a large heliotropic flower, which means it follows the sun throughout the course of the day. A sunflower will actually start out facing east, and then it will track the sun throughout the day, and it will face west by the end of the day. The collenchyma cells help to support the weight of the sunflower head, and that is where you find the collenchyma cells. Sclerenchyma cells are rigid due to thick secondary walls and containing lignin, and they have a structure. Lignin is the strengthening polymer. They are dead at functional maturity, and there are two types of sclerenchyma cells: sclerids, which are short, irregular in shape, and have thick lignified secondary walls, and fibers, which are long, slender, and grouped into strands. Okay, so the sclerids are specialized entirely for support and strengthening. Sclerids, which are boxier than fibers and irregular in shape, have a very thick, lignified secondary walls. Sclerids impart the hardness to nutshells and to seed coats, and the gritty texture to pear fruits. Fibers, which are also sclerenchyma cells, are usually grouped into strands and are long, slender, and tapered. Some of are used commercially, such as hemp for fibers for making rope, or flax fibers for weaving into linen. So these are fibers, sclerenchyma cells, that are found in plants that can be commercially useful. Now, okay. So these sclerid cells, when you eat a pear and it has that little bit of gritty texture in it, those are sclerid cells, the same kind of cells that make a walnut difficult to open. Okay, here are sclerid cells in a pear, the gritty cells that you chew on in a pear, and fiber cells from an ashtray. These are both types of sclerid cells. Water-conducting cells of the xylem are dead at functional maturity. So the tracheids and the vessel elements that form the basis of the conducting the water cells, they're not actually living cells. They are actually dead. There are two types of water-conducting cells. Tracheids are long, thin cells with tapered ends that move through water through pits. And vessel elements align end to end to form long micropipes called vessels. Tracheids occur in the xylem of all vascular plants. Vessel elements are common to most angiosperms, a few gymnosperms, and a few seedless vascular plants. Okay, there are two types of water-conducting cells, tracheids and vessel elements. They are tubular, elongated cells that are dead at functional maturity. Tracheids occur in the xylem of all vascular plants. In addition to tracheids, most angiosperms, as well as a few gymnosperms and a few seedless vascular plants, have vessel elements. When living, cellular contents of the tracheid or vessel element disintegrate. When the living cellular contents of the tracheid or vessel element disintegrate, the cell's thickened walls remain behind, forming a non-living conduit through which water can flow. Okay, here are the scanning electron micrograph of tracheids and vessels. You see the tracheids are the little small areas, and the vessels are the larger areas. This is what water moves through. This is part of the xylem. Sugar-conducting cells of the phloem are alive at functional maturity. So they differ from xylem in the way xylem moves water, phloem moves food. Xylem is dead, phloem is alive. So phloem are alive at functional maturity. And seedless vascular plants and gymnosperms, sugars are transported through sieve cells. Okay, in angiosperms, sugars are transported in sieve tubes, chains of cells called sieve tube elements. Sieve plates are the porous end walls that allow fluid to flow between cells along the sieve tube. Sieve tube elements lack organelles, but each has a companion cell whose nucleus and ribosomes serve both cells. Okay, so let's see. Do we go into some more detail? No, we don't. Okay. Okay, so here's a sieve tube element on the left, and okay, so phloem are alive at functional maturity. In seedless vascular plants and gymnosperms, sugars and other organic nutrients are transported through long, narrow cells called sieve cells. In the phloem of angiosperms, these nutrients are transported through sieve tubes, which consist of chains of cells that are called sieve tube elements or sieve tube members. Though alive, sieve tube elements lack a nucleus, ribosomes, a distinct vacuole, and cytoskeletal structures. The reduction in the cell contents enables the nutrients to pass more easily through the cell. The end walls between the sieve tube elements, called sieve plates, have pores that facilitate the flow of fluid from cell to cell through the sieve tube. Alongside each sieve tube element is a non-conducting cell called a companion cell, which is connected to the sieve tube element by numerous plasmodesmata. These nucleus and ribosomes of the companion cell serve not only to the cell itself, but also to the adjacent cell sieve tube element. In some plants, the companion cells in leaves also help load sugars into sieve tube elements, which then transport the sugars to other plants, other parts of the plant. Okay, so here are the plasmodesmata, which are small small holes in the cell. Here's the companion cell that is living alongside the living sieve tube element. The sieve tube element has these sieve plates between each sieve tube element through which the nutrient fluid can pass. Okay, so companion cells here are on the outside of the sieve tube. These are the companion cells here, and they basically run all the functions of the sieve tube element, and they do all of the work for it. And because the sieve tube itself is a living cell, but it's pared down to the bare bones minimum. It doesn't have a nucleus, doesn't have ribosomes, doesn't have any of the regular structures of a cell. Meristems generate new cells for growth and control of the developmental phases and lifespans of plants. A plant can grow throughout its life. This is called indeterminate growth. Indeterminate growth is enabled by meristems, which are perpetually undifferentiated tissue. Some plant organs cease to grow at a certain size. This is called determinate growth. Different meristems produce primary and secondary growth. There are two main types of meristems. Apical meristems, which form the growth on the tip of the plant, and lateral meristems, which form growth within the trunk of the plant. Okay, so here is the shoot tip apical meristem. Apical meristems add primary growth in length. Okay, there's apical meristems here on the end of this plant, and then also on the end of the roots. There are apical meristems on the end of the roots. And then there are, in woody plants, lateral meristems add secondary growth, increasing the diameter of the roots and stems. So here throughout say this was a tree, the vascular cambium and the cork cambium, these are areas that are growing and supplying lateral growth. These are the lateral meristems. So primary growth in stems. Is this a eudicot or a monocot? Well, we can tell it's a eudicot because it's got this structure to its vessels, organs, to the xylem and phloem. Up here at the top of the tree, it's got less complicated structure. It has the epidermis, the cortex. This is the ground tissue, the primary phloem, the primary xylem. These are the vascular tissue, and the pith, also part of the vascular tissue. Okay, so everything inside the vascular tissue is the pith, and everything outside the vascular tissue is the cortex. The secondary growth in stem includes the you got the same basic structure here, but it's more complicated. The pith is on the inside of the structure here, and the cortex is on the outside of the of the vascular tissue, and then between the vascular tissue, between the xylem and the phloem, there is growing, there is the increasing lateral meristem. This is increasing the girth of the tree, and this is where this growth occurs. Okay, so the cork cambium adds to the secondary dermal tissue, and the vascular cambium adds to the secondary xylem and phloem. Okay, here's our little tree. Apical meristems are located at the tips of the roots and shoots and the axillary buds of shoots. Apical meristems elongate shoots and roots in a process called primary growth. Lateral meristems add thickness to woody plants in a process called secondary growth. There are two lateral meristems, the vascular cambium and the cork cambium. The vascular cambium adds layers of vascular tissue called secondary xylem or wood and the secondary phloem. The cork cambium replaces the epidermis with periderm, which is thicker and tougher. In woody plants, primary growth extends the shoots and secondary growth increases the diameter of the previously formed parts. Okay, so if you look at the twig of a tree, like if you go out and find the twig of an oak tree or a maple tree on your walks, you'll see it's very clear where the apical bud is located. The bud scale are the little leaves surrounding the buds. The axillary buds here are located at the nodes. This is the tip of that branch is last year's growth. So this is from spring to fall of last year when it had leaves on that. It had leaves on that plant and helped it to grow. When that growth ended or began at the growth of the second year of the previous year's growth, and that growth began at that node of the bud scar of the previous year's growth. Okay, so these cells have these these little twigs have these bud scars in between them, and each bud scar is a year. So here's the initial year. There's a bud scar. The second year, there's a bud scar, and there's this year's growth. That's this last year's growth. And then here's the apical bud, and that's going to grow into a new twig, and then that new twig will be the next year's bud growth. It'll probably look basically the same as this. Just move it on down the branch. Branches will grow to as long as they can until they start to break off and die, or they could be affected by termites or ants or beetles. Any one of natural conditions can affect the growth of a branch. They can be blown off by wind or what have you. All these different aspects of nature function to basically naturally prune the tree so that trees will only grow to a certain height, but they continue to grow within that. They continue to grow new buds every year, new last year's bud scar on top of last year's bud scar. So all this growth of the tree is ongoing. Meristems give rise to initials called stem cells, which remain in the meristem. Derivatives which become specialized in mature tissues. Okay, so initials are like stem cells. They can become any different kind of tissue, and then a derivative is once that cell becomes specialized into a mature tissue. Cells of a gene expression and control of cell differentiation. Cells of a developing organism synthesize different proteins and diverge in structure and function even though they have a common genome. Cellular differentiation depends on gene expression, which is determined by position. Positional information is communicated through cell interactions. So for gene activation or inactivation depends on cell-to-cell communication. For example, Arabidopsis, which is a very well-known and highly studied form of garlic mustard, species that's very well known to scientists, the root epidermis forms roots hairs or hairless cells depending on the number of cortical cells it is touching. Okay, so this is the gene glabra2. Differential expression of a gene called glabra2 is required for the appropriate root hair distribution. Researchers have demonstrated that this requirement by coupling glabra2 to a reporter gene that causes every cell expressing glabra2 in the root to turn pale blue following a certain treatment. And the glabra2 gene is normally expressed only in epidermal cells that will not develop root hairs. Okay, so in this structure here, they've been able to dye this gene where glabra2 is expressed, and this will be these cells will remain hairless. In these other cells that do not have the glabra2 gene expressed, these cells will develop root hairs. In this way, it spaces out the need of the gene of the cells to differentiate and to form different root hairs. So you can see where the root cells will be sloughed off before the root hairs emerge here in this location, and these are the cortical cells. So this where they have a certain amount of contact between these glabra cells and the cortical cells, that will cause those to remain hairless. And where the cells are in between these cortical cells, when they're in between the cortical cells, that's where they will develop root hairs. Meristematic control of the transition to flowering and the lifespans of plants. Flower formation involves a transition from vegetative growth to reproductive growth. It is triggered by a combination of environmental cues and internal signals. Reproductive growth is determinate. The production of a flower stops by primary growth of that shoot. So once a flower is produced, it's determinate growth. That's distinct to indeterminate growth, which produces twigs and branches and buds and leaves. That is indeterminate growth. Flowering plants can be categorized based on the timing and completeness of the switch from vegetative to reproductive growth. So annuals complete their life cycle in a year or less. Biennials require 2 growing seasons to flower, and perennials can live for many years. So some bulb plants are like tulips are biennials. They require a season and then a second season to flower. And the perennials are like any of your bushes or woody plants. Plants that live for many years are perennials. Primary growth lengthens roots and shoots. Primary growth arises from the apical meristems and produces parts of the root and shoot systems. The root tip is covered by a root cap, which protects the apical meristem as the root pushes through the soil. Growth occurs just behind the root tip in 3 zones of cells: the zone of cell division, the zone of elongation, and the zone of differentiation or maturation. Okay, the root apical meristem produces all the cells of the root and the root cap. The most lengthening of the root occurs in the zone of elongation. In the micrograph, cells undergoing mitosis in the apical meristem are revealed by staining for cyclin, a protein that plays an important role in cell division. So here's these are the mitotic cells. They show you right at the tip of the root, the rootlet here. Okay, here's the where the root hairs root hairs are being formed. This is the zone of maturation or the zone of differentiation. This is where the root hairs are being formed, and this is where the relationship with the mycorrhizal fungi is occurring. Okay, the tip of your root is kind of pushing through the soil. This is the zone of cell division through here. It includes the root cap or apical meristem, and the root cap is like a bunch of dead cells that kind of protects the tip of the root from the action of it pushing through the soil. So the root grows, it actively pushes its way through the soil, and it works its way around rocks and other roots and what have you. And then if you've ever pulled up a plant, you can. It's pretty instructive as to what the root does. It grows through the soil and around other roots and around other obstructions, and finds its way into the best possible conditions. And then once it reaches those conditions, the zone of differentiation comes into play, and it matures and creates the root hairs. Okay, here's the mitosis of the cells occurring here in the zone of cell division right around the tip of the root. There's the mitotic cells. The primary growth of roots produces the epidermis, the ground tissue, and the vascular tissue. In angiosperm roots, the stele is a vascular tissue, a vascular cylinder. In most eudicots, the xylem is a star-like appearance with the phloem between the arms. In many monocots, the core of parenchyma cells is surrounded by rings of xylem and then phloem. Okay, so here on the left we have a root with a xylem and a phloem in the center, typical of eudicots. In the roots of typical gymnosperms and eudicots, as well as some monocots, the stele is a vascular cylinder appearing in the cross section with the lobed core of xylem and phloem beneath the lobes. Okay, so here's the xylem and phloem that's occurring in the center of this eudicot root structure. And in the monodicot root structure, roots with a parenchyma in the center are typical of monocots. The stele of many monocot roots is a vascular cylinder with the core of parenchyma surrounded by a ring of xylem and a ring of phloem. Okay, here's the epidermis, the cortex, the endodermis. The vascular cylinder is inside the endodermis. The pericycle is part of that ground tissue, part of that vascular tissue. The xylem and the phloem, other parts of the vascular tissue inside deep inside that root. Okay, here on the monocots, it's dominated by the vascular tissue, particularly by the parenchyma on the inside of the core of parenchyma cells. There's the endodermis and the epidermis, the cortex. Okay, these are all parts of the monocot cell root structure. Ground tissue, mostly parenchyma cells, fills the cortex, the region between the vascular cylinder and the epidermis. The innermost layer of the cortex is called endodermis. The endodermis regulates passage of substances from the soil into the vascular cylinder. Lateral roots arise within the pericycle, the outermost cell of the cell layer in the vascular cylinder. Okay, here's an emerging vascular lateral root inside of a main root structure here, and we can see the pericycles surrounding that area, a vascular cylinder, and the cortex. Okay, a lateral root originates from in the pericycle, the outermost layer of the vascular tissue, a vascular cylinder of the root, and grows throughout the cortex and epidermis. In this series of light micrographs, the view of the original root is cross-section, while the view of the lateral root is in the longitudinal section. Okay, so Here you can see the root starts here just outside the pericycle, and then it develops into greater and greater strength, and then it grows out and eventually becomes the lateral root. So you can see the lateral root, the emerging lateral root, growing from the center of the root itself, the pericycle, and then it grows outwardly through the epidermis, and it creates its own structure. All right, this is all mitosis. This is where the zone of cell division is occurring in the lateral root here, and once it reaches maturity, then around the side here, it will develop root hairs in that zone of differentiation. Okay, this is a typical eudicot. Primary growth of shoots. A shoot apical meristem is a dome-shaped mass of dividing cells at the shoot tip. Leaves develop from leaf primordial projections along the sides of the apical meristem. Shoot elongation results from the lengthening of internode cells below the shoot tip. Okay, so here is the, it looks kind of like the horns of a bull. The leaf primordia arise from the flanks of the dome of an apical meristem. This longitudinal section is a shoot tip of coleus. I'm not sure what kind of plant coleus is, but you can see here this is like inside the bud of the branch tip, and then this is what it looks like. The leaf primordia, these are eventually going to become leaves. This would be the blade of the leaf, and this is going to turn into the petiole, and it's going to attach to the branch there. Okay, so here's a young leaf here on the outside of the shell, and inside this is the developing vascular strand, and inside here this is where this would become the internode. So this would be the apical meristem, the node, and then the internode until the axillary bud meristems, also another form of apical meristem down here. Okay, this is going to form these structures here, and then this is where the this is where the main bud growth is occurring. So when you take those twigs down in the springtime and you look at the end of the buds, this is what it looks like inside them. Axillary buds are dormant due to apical dominance and inhibition from active apical bud. When released from dormancy, the axillary buds give rise to lateral shoots. So the apical those apical buds, they can just okay, if there's another bud that's grown out before it, then it inhibits the growth of that bud. If that bud gets broken off, it gets eaten by a giraffe or broken away by the wind, it can release that potential for that axillary bud to give rise to lateral shoots. Tissue organization of leaves. The epidermis in leaves is interrupted by stomata, which allow for carbon dioxide and oxygen to exchange between the air and the photosynthetic cells of a leaf. Each stomatopore is flanked by two guard cells, which regulate its opening and closing. The ground tissue in a leaf is called mesophyll. It is compared of parenchyma cells sandwiched between upper and lower epidermis. Okay, so here's the structure of, this is what a leaf looks like. Surrounding the outside of the leaf is the dermis shell, the dermis tissue. This is the cuticle, which is like a waxy formation that helps to prevent water from evaporating from the surface of the leaf. The cuticle it's obviously more developed in drier plants that have a lot of in a drier environment. But the waxy cuticle can also be highly developed in rainforest plants because it helps to defend the leaves against certain fungus that may inhibit the ability of the plant to photosynthesize. So there's different advantages to that structure. Okay, the cuticle is the surface of the leaf. Sclerenchyma fibers are forming on the inside of the shell. This is where the palisade mesophyll occurs. This is where the chloroplasts are mostly concentrated. The spongy mesophyll is below the palisade mesophyll. It's less structured. It's on the underside of the leaf. It probably forms some form of structural reason for that. The spongy mesophyll also has chloroplasts in it, but it's not as highly structured as the palisade mesophyll. There's a vein which supports the xylem and the phloem, the vessels, the vascular tissue of the leaf. So these are the xylem is bringing water up from the roots, and the phloem is taking carbohydrates back down through the rest of the plant and into the roots. Okay, so this is the bundle sheath cell. Forms the vein, forms the exterior of the vein, and this is on page 565. If you want to get a more closer understanding of this, these are the stomata, the stoma here, which open up and allow for gas exchange from the cell, from the leaf to the atmosphere, and vice versa. So carbon dioxide comes in and oxygen goes out. And at night during the dark cycle, you remember you all studied photosynthesis in Bio 101. During the dark cycle, oxygen comes in and carbon dioxide goes out. Okay, so here are the guard cells of a stomata here in the surface view of a spiderwort, and here's the cross section of a syringia. It shows the veins here, air spaces between the spongy mesophyll and the palisade mesophyll as they're more highly structured, and the upper epidermis, and the cuticle is going to be found on top of that upper epidermis. Okay, so here's a better view of it. Okay, just figure 8. So this is figure 28.17 in your textbook. It provides an overview of the leaf structure. The epidermis is interrupted by pores called stomata, singular stoma, which allow exchange of carbon dioxide and oxygen between the surrounding air and the photosynthetic cells inside the leaf. In addition to regulating carbon dioxide uptake for photosynthesis, stomata are major avenues for the evaporative loss of water. The term stoma can refer to the stomatal pore or the entire stomatal complex, consisting of a pore flanked by two specialized epidermal guard cells, which regulate the closing and opening of the pore. Okay, we'll discuss the stomata in greater detail later. The mesophyll of Eudicots has two layers. The palisade mesophyll of the upper part of the leaf consists of elongated cells for photosynthesis, and the spongy mesophyll of the lower part of the leaf consists of loosely arranged cells, primarily for gas exchange. The vascular tissue of each leaf is continuous with the vascular tissue of the stem. The veins of the leaves are vascular bundles which function in fluid transport and provide structure to the leaf. Each vein in a leaf is enclosed by a protective bundle sheath. Tissue of organization of stems. Stems are covered in epidermis and contain vascular bundles that run through the length of the stem. Lateral shoots develop from axillary buds on the stem surface. In most Eudicots, the vascular tissue consists of vascular bundles arranged in a ring. Okay, this is on the left is a cross section of a stem with vascular bundles forming a ring typical of eudicots, and on the right is a cross section of a stem with a scattered vascular bundles typical of monocots. Okay, this just shows the different parts and parcels of. You should be able to recognize everything here: the xylem and the phloem, the vascular bundles, the sclerenchyma, the fiber cells, the ground tissue connecting the pith to the cortex, the pith, the cortex, the vascular bundle, the epidermis. Okay, all these different parts you should be able to recognize and understand, because we've gone through this in this lecture. So this is just a review of the material that we've already covered in the lecture. In most monocot stems, the vascular bundles are scattered throughout the ground tissue rather than forming a ring. Okay, so monocots seem to be they're differently organized than eudicots. Eudicots, when you see a highly organized stem cut across, you can tell that that's a eudicot. Monocot stems, vascular bundles are scattered throughout the ground tissue. Okay, so that's a characteristic that when we looked at some of these differences between eudicots and dicots in those in the models they use. The models they use are mostly eudicots, and they have a much more structured vascular bundles. Secondary growth increases the diameter of stems and roots in woody plants. Secondary growth is a characteristic of gymnosperms and many eudicots, but not monocots. Okay, there aren't any really woody monocots, except for the fact that palm trees are monocots, and but they're not really woody. So palm trees do have a certain amount of secondary growth, but I don't know why they don't talk about that here. But let's just, for the purpose of this discussion, we're just going to go with what the book says, which is secondary growth is characteristic of gymnosperms and eudicots, but not monocots. Secondary growth occurs in stems and roots of woody plants, but rarely in leaves. The secondary plant body consists of the tissues produced by vascular cambium and cork cambium. In woody plants, primary growth and secondary growth occur simultaneously, but in different locations. Okay, we're going to break this down a little as we go through this here. But at the top you see basically as you go down the structure of the tree, you're going to get more bark and more cork. You get more lateral growth outward, and the cork and the vascular cambium, they're all small. They're reduced in size at the tip towards the apical meristem, and then as you get further away from the apical meristem down to the trunk of the tree or the more substantial part of the branch, then you're going to get different structures. You're going to get more complex structures in here. Now we're going to go through this. I believe they're going to take us through this in a little more detail. Okay, so primary growth, this is the activity from the apical meristem is nearing completion, and the vascular cambium has just formed. The vascular cambium is that red line here. Although primary growth continues in the apical bud, only secondary growth occurs in this region. The stem thickens as the vascular cambium forms, and the secondary xylem forms a secondary xylem to the inside and the secondary phloem to the outside. So the xylem is getting split off to the inside, and the phloem is getting split off to the outside. Some initials of the vascular cambium give rise to vascular rays. So initials are little structures that grow into these vascular rays. So the vascular ray is going to connect the the secondary phloem and the secondary xylem. So it provides a structure. The vascular ray provides a structure between the secondary xylem and the secondary phloem. So there's ability to communicate. You can get fluids back and forth across those. Okay, then there's the first cork cambium and the cork. The cork is basically what underlies the bark. And I don't know if you're aware of this, but if you uncork a bottle of wine, the cork from that bottle of wine comes from a particular type of oak tree known as a cork oak, and they're grown mostly in Spain. So the cork oak grows a really thick cork underneath its bark, and it can be you can harvest that oak, harvest that cork from the oak, and then they mill it into corks, which is the cork for your wine bottle. Okay, some initials of the vascular cambium give rise to vascular rays. As the vascular cambium's diameter increases, the secondary phloem and other tissues external to the cambium can't keep pace because their cells no longer divide. As a result, these tissues, including the epidermis, will eventually rupture. The second lateral meristem, the cork cambium, develops from parenchyma cells into the cortex. The cork cambium produces cork cells, which replace the epidermis. Okay, in year 2 of secondary growth, the vascular cambium produces more secondary xylem and phloem. The cork cambium produces more cork, and the stem's diameter increases to the outermost tissues of the exterior of the cork, where the cambium rupture and are sloughed off. In many cases, the cork cambium reforms the deeper in the cortex. When none of the cortex is left, the cambium develops from phloem to parenchyma cells. Each cork cambium and the tissues it produces form a layer of periderm. Bark consists of all tissues exterior to the vascular cambium. So everything exterior to the vascular cambium, which is this here, the vascular cambium. This is everything. Following that is the bark. Okay, so all this whole structure here is the bark. Now, it's actually possible to kill a tree by doing what they call girdling it, which is cutting off everything surrounding the bark. If you cut down into about an inch or two surrounding the cork, if you cut underneath the cork of the tree, you can actually kill the tree by disrupting the flow of xylem from the tree trunk to the branch. It's believed that this is what people do under really primitive circumstances when they want to fell a tree. They cut a circle that's called girdling the tree. They girdle the tree, and then the tree eventually dies from lack of phloem to the top of the tree, and then the tree falls over. This can take many months or even a year or two. So it's a very slow way of clearing a field for pasture. But if you have no other means to do it, it's, especially in primitive cultures, this is the way that trees are cut down. Okay, so here's the progress of the secondary growth can be tracked by examining the sections through sequentially older parts of the stem. Okay, so here are little parts like the vascular ray. Here's the cork and the bark. This is the whole outer part of the structure. Everything outside of the secondary phloem. The secondary phloem is just in here. Okay, and then here is 1 year's worth of growth. There's 2 years worth of growth. And when you count the rings on a tree, that's every year of growth around these strings. So this stem here, or this tree, is 1, 2, 3 years old. That is how you perform dendrochronology, which is the science of aging trees. Everybody can do it. Everybody's done this in Boy Scouts or Girl Scouts. It's very easy. You count the rings of the tree, and that's how old they are. Okay, well, those rings are, right, that is the growth ring, the secondary, the vascular cambium. The vascular cambium and secondary vascular tissue. The vascular cambium is a cylinder of meristematic cells of one cell layer thick. In cross sections, the vascular cambium is a ring of initials or stem cells. The initials increase the vascular cambium's circumference and add secondary xylem to the inside and secondary phloem to the outside. Okay, so although the secondary xylem and phloem are shown being added equally, cambial initials usually produce more xylem. Okay, so these structures here, this is showing where the growth is occurring. A cambium initial can also divide to form an initial or in either secondary xylem or cell. The cambium initial can divide to form two cambium initials, increasing in the circumference of the vascular cambium. Okay, so so this is occurring inside the vascular cambium. Here's the cambium here in the central, and then it's producing can divide to form a phloem here or a xylem on the inside, and so it keeps producing that cambial vascular cambium is an initial like stem cell. So the initial stem cells are located here on the cambium, and they are producing more and more phloem on either side, on one side, and more xylem on the inside. So phloem on the outside and xylem on the inside. Okay, so after 2 years of growth, they have most of the thickening is from the secondary xylem. Elongated, parallel-oriented initials produce tracheids, vessel elements, fibers of xylem, and sieve tube elements and companion cells, axially oriented parenchyma, and fibers of the phloem. Shorter, perpendicular oriented initials produce vascular rays, radial files of parenchyma cells, and connect secondary xylem and phloem. Secondary xylem accumulates as wood and consists of tracheids, vascular vessel elements only in angiosperms, and fibers. Early wood formed in the spring has thin cells to maximize water delivery, and late wood formed in the late summer has thick-walled cells and contributes more to stem support. In temperate regions, the vascular cambium of perennials is inactive throughout the winter. Okay, so tree rings are visible where late and early wood meet and can be used to estimate the tree's age. Dendrochronology is the analysis of tree ring growth patterns and can be used to study past climate change. So where these tree rings are far apart, we can infer that that was a wet year, and when they're closer together, we can infer that that was a dry year. As a tree or woody shrub ages, the older layers of secondary xylem, the heartwood, no longer transport water and minerals. The outer layers, known as the sapwood, still transport materials through the xylem. Only secondary phloem transports sugar. Older secondary phloem sloughs off and does not accumulate. Okay. All right, so here is a structure of a tree. It shows you the growth ring between years, a vascular ray, which joins the xylem and the phloem. The heartwood, which is no longer functions to pass food and water. The sapwood, which is where most of the activity of the xylem is occurring. The vascular cambium, this is where the plant stem cells are located, where the initials are located, forming the vascular cambium. And the secondary phloem, this is where most of the phloem activity, the transport of food, liquid food, is occurring in the secondary phloem. And this is the layers of the periderm. So everything outside of the vascular cambium is the bark, and the periderm is the skin of the tree. The cork cambium and the production of the periderm. The cork cambium gives rise to cork cells that accumulate in the exterior of the cork cambium. Cork cells deposit waxy suberin in their walls and then die. Periderm consists of the cork cambium and the cork cells it produces. Periderm functions as a protective barrier, impermeable to water and gases. Bark consists of all the tissues external to the vascular cambium, including the secondary phloem and the periderm. Lenticels in the periderm allow for gas exchange between the living stem or the root cells and the outside air. Remember we talked about the pneumatophores or the lenticels, the structures of the mangrove swamps. Okay, those have lenticels that allow the exchange of gas between living cells and root cells into the outside and the outside air. Okay, this is an experiment where they took maple leaves they transplanted them in either Rhode Island or Florida, and then they compared the average area of a single tooth on that leaf versus the number of tooths on those leaves. You're supposed to make a bar graph with this. I think this is a exercise on page 560. Okay, so the question is, are these differences related to genetic differences, or are they related to environmental differences? Well, if you look at Florida, the ones grown in Florida and the ones grown in Rhode Island, here are maple trees. Maple trees grown in Florida have a lot of a large tooth area. Take that same tree and grow it in Rhode Island, and it has less tooth area. Then you compare the number of trees from Florida grown in Florida. They have less leaf area, and you take that same tree, seeds from that same tree, and you grow it in Rhode Island, and it has more leaves per tooth area. So it's got, if the Florida, anyway, what you're looking at is the characteristics of the leaf structure. It does seem to vary based on environment rather than genetics. You can get into more detail in this on page 560. Okay, here's our schematic of our plant. How does the plant work? Overview of primary and secondary growth. The apical meristems add primary growth and grow in length, and the lateral meristems add, that's the cambium, that adds secondary growth. Okay, so we're looking at the inside here. We've got the cork cambium and the vascular cambium. Those are two areas that promote growth through their stem cells. Okay. This is a image of woody eudicot. It says here, label the growth ring, late wood and early wood, and the vessel element, and draw an arrow to in the direction of the pith to cork. Okay, so you have to figure out how this is working. This is an exercise on page 570 in your textbook.